Okay, welcome back, everybody. It's 1 p.m. Central, and we will start with our afternoon session. This session has three parts. We have two additional speakers during this section, and then we will have a 2 hour long panel where I think we will get to ask all of the questions that we want, and some of the questions that we have not been able to answer live after each of the panelists. We will be considering some of those at the end of the panel discussion, probably 20 to 30 minutes after the discussion. So welcome back, everybody. Our next presenter is Dr. Matt Regan, and he's going to be talking about hibernation and gut microbiome. And for the ones of you who were able to attend a few weeks ago our microbiome workshop, you will see that there's some similarities here, and you might be able to connect the dots here. So, Dr. Regan, whenever you're ready, you can share your screen, and the stage is all yours.
All right. Thanks, Manuel. Can you hear me?
Yes, I can.
All right. So thanks for the intro. And thank you very much for inviting me to be part of this really interesting event. It makes me excited to know that we're thinking about grand challenges like this one. And I also want to say that I'm really honored to be included among such a great list of speakers. So thank you very much. So today I'm going to talk about hibernation and the gut microbiota. And I'm going to split this talk into three parts. So first, I'm going to introduce the gut microbiota, and I'm going to talk about how it is affected by hibernation. Second, I'll talk about the reverse side of that coin, which is how the gut microbiota affects hibernation. And then in the third part of the talk, I'll talk a bit about how I believe we should be thinking about the gut microbiota with respect to applying synthetic torpor, to use Matteo's term, to long-duration human spaceflight scenarios. And this will, of course, be from my perspective. I'm an animal physiologist who focuses on various aspects of metabolic depression, including hibernation. But also, I'm a really big space enthusiast who, as Manuel mentioned during the intro, I've collaborated with scientists at NASA and elsewhere on projects related to this topic. So to start, we'll start by talking about the gut microbiota generally. So as you probably know, our bodies are comprised not only of eukaryotic human cells, but also of prokaryotic microbial cells, particularly bacterial cells. In fact, our bodies contain more bacterial cells than human cells, and way more bacterial genes than human genes, which, when first learned, can cause, the contemplative human to spend a little more time pondering just who they really are. Now, the majority of these bacteria live in our GI tracts, particularly in the large intestine, where they live in relative harmony with us, their hosts. But the host-microbe relationship is complex, and work over the past 10 or 15 years in particular has started to illuminate the amazing ways that the host can influence the gut microbiota, and perhaps more interestingly, how the microbiota can influence the host. So generally speaking, and I'll use a little cartoon to illustrate this point, gut microbes, which are these various ovals here, gut microbes communicate with the host through the chemicals that they produce, namely small molecules called metabolites, which are these black dots here. And via the absorption of these metabolites and their subsequent distribution throughout the body, gut microbes can influence a variety of processes within the host, ranging from metabolic fuel use to immune function to behavior. And because of these many and varied influences on the host, some experts are starting to consider the gut microbiota another organ of the body. Now, thus far, I've been speaking with respect to humans, but of course, other animals contain gut microbes too, including hibernating animals. And there's been speculation on the possible role of the gut microbiota in hibernation floating around in the literature since at least the 70s. This is the first or the earliest paper that I know of, which coincidentally has a spaceflight connection here. But the actual study of this role has really only intensified over the past 10 or so years. It's focused mainly on bats, bears, and hibernating ground squirrels. And for the most part, these studies have investigated what happens to the gut microbiota over the hibernation season, which is to say how hibernation affects the gut microbiota. And by and large, what these studies have revealed is that over the hibernation season, the overall abundance of and the diversity of gut microbes decreases. So with respect to abundance, and taking a study on the Arctic ground squirrel here as an example, the total abundance of gut bacteria decreases about tenfold by late in the hibernation season relative to summer time, which we see in this highlighted column here. This is a log scale, as you can see. Similar results have also been shown in the 13-line ground squirrel using a different technique, which suggests that decreases like this in overall abundance of gut microbes may be a general trend among fasting hibernators. Next, with respect to microbial diversity, we have a bit more data to draw from, and the results are consistent enough for us to draw a few general conclusions. So first, during hibernation, the diversity of microbes present in the gut decreases, as shown by these different measurements of alpha diversity here in the 13-line ground squirrel, Arctic ground squirrel, and brown bear, where you can see that the winter hibernating groups, shown here, here, and over here, have a lower overall diversity of microbes than the summer groups. Now, the exception to that is this post-hibernation group in the Arctic ground squirrel study here, but I should clarify that what post-hibernation means in their context. It was 3 days after the animals had emerged from hibernation and before they were given any food to eat. And we'll talk more about food and the influence of food in a moment. Second, during hibernation, different bacterial phyla are affected in different but consistent ways. So as shown on these relative abundance figures here, for the 13-line ground squirrel and the Arctic ground squirrel, bacteria belonging to the Bacteroidetes phyla and the Broca microbia tend to increase in relative abundance in hibernation relative to summer, whereas those belonging to the Firmicutes phyla tend to decrease in relative abundance. And these trends are also true for, generally true for the horseshoe bat and for the brown bear. So this is interesting, because despite the fact that these are different species that live in different environments, there's some consistency to their gut microbiotas and how it changes over the hibernation season. So much so, in fact, that it's been posited that perhaps there is a core gut microbiota that exists among obligate hibernators, like the 13-line ground squirrel and the Arctic ground squirrel that we see here. So the question, therefore, is what's driving this consistency in gut microbiota changes? And one of the leading theories, in fact, the leading theory, is that it's diet, or more specifically, a lack of diet. So during hibernation, obligate hibernators, like the ground squirrels, voluntarily fast for the duration of hibernation. This is the whole point of hibernation, after all, which is a strategy for dealing with a seasonal, usually a wintertime, lack of food. And it's a really effective strategy, but it does have some off-target effects, including the gut microbiota. So this is because many gut microbes rely on food ingested by the host for their own nourishment. So when the host fasts, as it does during hibernation, the food source for many gut microbes disappears. And I'll illustrate what I mean with here we have a schematic of the squirrel GI tract and a blown up portion of the cecum, which is right here. So as I said, many gut microbes rely on food flowing through the intestines for their own nourishment. And when that food stops flowing, as it does during hibernation, many of these microbes starve and die. Now, importantly, not all microbial species rely on the host diet for their nourishment. Some species can, or in fact need to, use as their carbon source mucin proteins that are secreted by the host into this mucus layer. And this happens throughout hibernation. So as this first group of microbes starves and dies, they free up space for the mucin degrading bacteria to occupy. And this idea is consistent with some of the general trends that we see in the way the gut microbiota changes in hibernators during hibernation. Bacterial phyla that increase in relative, or that decrease in relative abundance over hibernation, such as the Firmicutes here, they rely primarily on dietary source polysaccharides, whereas those that increase in abundance, such as the Bacteroidetes and Broca microbia, they're able to degrade host-derived compounds such as these mucin proteins. So another way to confirm the predominant role of diet in these hibernation-related microbial changes is to see what happens in food-storing hibernators. So bears, bats, and ground squirrels, they all fast during hibernation, but other species such as hamsters and chipmunks, they store food in their hibernacula and they nibble on it when they periodically arouse from torpor throughout the hibernation season, as we saw during Dr. Martin's talk. Now, our diet hypothesis would predict that in food-storing hibernators, gut microbial changes during hibernation will be less pronounced than they are in fasting hibernating species. And in fact, this is what we see. So these are for Syrian hamsters, and hamsters that were hibernating with food stores exhibited no changes in their gut microbial abundance or diversity, which is shown in these two columns here, relative to non-hibernating hamsters. While interestingly, summer active hamsters that were fasted for a mere 4 days exhibited gut microbial changes that resembled those that we see in our hibernating ground squirrels, shown here where there's a decrease in abundance, and this middle column here where the Firmicutes decreases and Verocamicrobia increases. And I think that this is an important point when we ponder just how to use synthetic torpor in spaceflight scenarios. And I'll get back to this idea in the third part of my talk. So to summarize this part of the talk, in fasting mammals and fasting hibernating mammals, generally speaking, gut microbes that rely predominantly on the host diet decrease in relative abundance throughout hibernation, whereas those that can degrade host-derived substrates such as mucin proteins increase in relative abundance over hibernation. And overall, we see about a 10-fold reduction in the overall abundance of gut microbes by late winter or late in the hibernation season. There are probably some other factors that play a role too, such as the drop in body temperature that we've talked about this morning, but there just hasn't been much work done on that at this point. So the question is, what does this all mean for the animal? In other words, how do these gut microbial changes impact hibernation? Well, hibernation biologists and microbiologists are really just starting to begin to address and understand this. In fact, even in non-hibernating species, we're still in the early phases of understanding just how gut microbes can influence the host. So given this, what I'll do is I'll first speculate on how the gut microbiota may influence certain hibernation traits. And then I'll talk about a project that I'm currently working on with Hannah Carey at University of Wisconsin Madison, that to my knowledge is the first study to really nail down the functional role of the gut microbiota in hibernation. So many of the gut microbiota's potential roles in hibernation physiology have to do with its production of short-chain fatty acids. So short-chain fatty acids affect a variety of processes in the gut and elsewhere, and they are sourced primarily from the gut microbiota. During hibernation, the cecal concentrations of the major short-chain fatty acids, acetate, propionate, and butyrate, they all decrease, but they do so to different extents. So butyrate here, you can see, falls to pretty much negligible levels during the hibernation season, whereas acetate here, it decreases too, but it remains in appreciable levels during the hibernation season. These changes may be involved in some hibernation-related changes that we know about. For example, there's a general mucosal atrophying during hibernation, including the epithelium. Now, epithelial cells use butyrate as their preferred fuel source. Therefore, the greatly reduced butyrate levels that we see during hibernation may be involved in this intestinal atrophy. Another example, the heart and the brain of hibernators use ketones as their preferred fuel source. And acetate can be converted into ketones in gut epithelial cells and in the liver. Therefore, the relatively high concentrations of acetate that we see in the cecum during winter may be used in part to help power the heart and the brain during hibernation. Another example, gut-derived serotonin production in mice, which is involved in glucose homeostasis, has recently been linked to short-chain fatty acids produced by the gut microbes. And this may be the case with hibernators too, with a possible involvement of seasonal fuel switching that is characteristic of hibernators. In the summertime, hibernators burn primarily glucose, whereas when they enter the hibernation season, they switch to burning lipids. But at this point, these gut microbiota roles, they're all speculative. This is a very recent area of hibernation research, and we just don't have any more data yet at this point to do much more than speculate. But probably the best evidence we have on the gut microbiota's role in hibernation is a project that I'm currently working on that I mentioned earlier. And this project has to do with protein synthesis during hibernation. So as we've established, obligate hibernators like the 13-line ground squirrel that I'll talk about for this part of the talk, they fast for the entirety of the hibernation season, which again is a great solution to a food scarcity problem. But it does deprive the squirrel of an exogenous source of nitrogen, and this could consequently jeopardize the squirrel's ability to synthesize new proteins. And this could be problematic, especially considering that the squirrel's reproductive season, which is a physically active time of year that would benefit from proper muscle function and protein synthesis, this reproductive season follows immediately on the heels of the squirrel's emergence from hibernation in the springtime. However, we know a couple of things. So first, hibernators like the ground squirrel tend to be quite resistant to muscle atrophy despite their many months of inactivity during the hibernation season. And second, data from Sandy Martin's group from a few years ago showed that protein synthesis rates in the muscle of 13-line ground squirrels actually increased towards the end of hibernation, almost as if in preparation for this emergence into the reproductive season as they emerge from hibernation. So the question, therefore, is how do hibernators build protein in the absence of dietary nitrogen? And this is where the gut microbiota comes in. So one longstanding hypothesis posits that hibernators, like ruminant animals, recycle the nitrogen in the urea they produce through a gut microbe-dependent process called urea nitrogen salvage, or UNS, as I may abbreviate it. Now, the published evidence for this process actually existing in hibernators is very scant, but we can look to how this process works in ruminants to have some idea of how it may work in our hibernating ground squirrels. So here we have our simplified diagram again: intestinal lumen, epithelial cells, blood. And here we have urea. So urea is produced by the squirrel primarily through the catabolism of proteins, and there's urea in the blood. And some of this urea could be transported into the intestinal lumen via urea transporters, or UTBs, that are present on these epithelial cells. And once in the intestine, that urea is surrounded by microbes, some of which may be urealytic, meaning they possess an enzyme called urease that is capable of hydrolyzing that urea into CO2 and ammonia. Now, importantly, the squirrel itself, like all vertebrates, does not possess the genes for urease. So it itself cannot hydrolyze urea in this way. Only the microbes can do this. And as they do, they produce the CO2 and ammonia. And so there are different possible fates for these two products. So for CO2, it can be either excreted by the squirrel, or it could be fixed possibly by other microbes and converted into organic compounds that could be used by those microbes, or possibly absorbed by the squirrel, thus recycling urea carbon. And ammonia, it can be taken up by the squirrel as is, or it can be converted by microbes into amino acids, which could be used by the microbes and or absorbed by the squirrel. Once in the squirrel, there are different fates for these compounds again. So the ammonia could be converted back to urea in the liver, or it could be converted into amino acids. And for the amino acids, they could be used as is, or they could be converted into other amino acids or proteins in the liver, or used in other peripheral tissues. The main point here is that these nitrogen atoms are urea derived, and they can be reincorporated into the hibernator's protein pool. And this process depends entirely on urease, which is to say it depends entirely on gut microbes, because vertebrates like the ground squirrel do not possess the genes for urease. So as I said earlier, there's very little evidence for this process existing in hibernators. So what we're doing is we're investigating major steps of this mechanism for evidence of them occurring in our hibernating ground squirrels. And we're doing this as a function of season. So we have 3 seasonal groups. We have a summer active group. We have an early winter group, which is 1 month into hibernation. And then we have a late winter group, which is 3 to 4 months into hibernation. And within each of these seasonal groups, we have 2 additional groups. So one with a naturally intact gut microbiota, and another with a depleted microbiota, which we accomplished using an orally administered cocktail of antibiotics. So for the sake of time today, I'm going to go through some of the results that I'm going to mention here pretty quickly, but I'm happy to discuss details of this project in more depth with anyone, either during the question session or via email later on if you'd like to learn more. Okay, so first, we found that winter hibernating squirrels express greater abundance of urea transporter in their ceca than did summer active squirrels. This suggests that hibernating squirrels may have a greater capacity to move urea from the blood and into the gut lumen, where the microbes are and where urea hydrolysis presumably takes place. Next, on the topic of urea hydrolysis, we've made real-time measurements of microbial urealytic activity using stable isotope labeling and a technique called cavity ring down spectroscopy, or CRDS. And so briefly, we inject squirrels with a mass-specific doubly labeled, or bolus of doubly labeled urea here. And then we collect the CO2 that they exhale and send it through a CRDS unit. And what the CRDS unit does, it measures the ratio of C13 CO2 to C12 CO2 in the squirrel's breath. Remember that the hydrolysis of urea yields ammonia and CO2. We call this ratio the delta C13 value. And so the higher this ratio is following the injection of the urea here, the greater the urealytic activity. And so looking at some of these results, we can see that there is a significant increase in breath delta C13 value in all 3 seasons for our squirrels with the intact microbiotas, which are the solid symbols that you see here. And we know these results are microbial in origin because the squirrels with depleted gut microbiota, shown in the open circles here, show minimal response following the injection of that urea, which took place right here at time 0. So, oh, and I should say that one of the other things that we see is that there is a pretty significant seasonal effect, whereby summer squirrels show significantly higher urealytic activity than early winter or late winter squirrels. And we believe this is due simply to the overall decrease in gut microbial abundance that I mentioned earlier in the talk. So what these results demonstrate is that hibernators' gut microbes can hydrolyze urea. And so the question now is the squirrel benefiting in any way from this urea hydrolysis? And we're addressing this a couple of different ways, both of which are based on the fact that we've treated these squirrels with doubly labeled urea. So first, we're measuring urea nitrogen incorporation into the small molecules or metabolites of different tissues of the squirrel using N15 specific NMR. And then second, we're measuring urea nitrogen incorporation into the protein contents of different tissues of the squirrel using a technique called isotope ratio mass spectrometry. So increases in the N15 incorporation in either of these molecules indicate or indicative of urea nitrogen incorporation. We're currently in the process of analyzing the metabolomic data. So for today, I'll just talk about the protein data. So starting with the liver, we see that there is a significant incorporation of N15 into liver protein, and that this is a likely microbial in origin because the squirrels with the depleted microbiota show minimal incorporation of N15. And interestingly, this is sort of consistent with those results of Sandy Martin's group that I mentioned earlier where they saw an increase in protein synthesis towards the end of hibernation, because you can see that this response is actually, or this incorporation of N15 is actually greater in hibernation than it is during summertime. And next, looking at muscle protein, we see by and large the same results here, albeit at a lower overall magnitude of N15 incorporation. So what these results mean is that hibernators can harness their gut microbes to recycle urea nitrogen back into their protein pools during hibernation when they lack an exogenous source of this important molecular building block. And for us, this is really exciting because it's the best evidence yet that the gut microbiota plays a direct role in the metabolic phenotype of hibernation. So to summarize this part of the talk, we can see that the gut microbiota benefits protein synthesis during hibernation via this urea nitrogen salvage mechanism. And it may affect other hibernation-related phenotypes too, but a lot of additional work needs to be done to assign any causality to these potential links. So this brings us to the third part of the talk, which is what all of this means in the context of synthetic torpor application to human spaceflight. Now, there will be some speculation from me here over the next few minutes, mainly because there just hasn't been the research done to do much more than speculate at this point. However, to guide that speculation, I think it really comes down to these two questions here, which relate to these two sections, the first two sections of the talk. So first, how will synthetic torpor affect the gut microbiotas of crew members? And then second, how will the gut microbiota affect synthetic torpor in crew members? So let's start by talking about the second one here. And I'll begin with this revealing observation from our lab. When we deplete the gut microbiotas of ground squirrels using antibiotics, those squirrels hibernate pretty well, just like the squirrels with naturally intact microbiotas. They go into torpor at the same time. They arouse throughout hibernation at about the same rate. They emerge from hibernation with no obvious detriments. What this suggests, at least at our current somewhat coarse level of analysis, is that the gut microbiota may not be critical to the induction of, or maintenance of, or recovery from torpor. And this may mean that we need not worry too much about the gut microbiota when developing methods and techniques for inducing synthetic torpor in humans, including aboard spacecraft. But of course, as we learn more about the gut microbiota's role in these metabolically depressed states, this conclusion may need modifying, as conclusions sometimes do. Manuel, I just have a couple more minutes left. But for now, I think the flip side of this coin is a little more concerning. That is, how might synthetic torpor affect the gut microbiota? And ultimately, how might this affect the health of crew members? Well, I believe the answer to that question lies mainly in the way we plan to use synthetic torpor. And there are different ways we can do this. And I'll illustrate those ways with a hypothetical graph that plots metabolic rate as a function of time. So one variable of synthetic torpor use is the depth of metabolic depression. Theoretically, synthetic torpor could be a deep state of metabolic depression, akin to what we see in our hibernating ground squirrels. Alternatively, it could be a relatively shallow state, akin to slow wave sleep that we talked about earlier this morning. Or it could be anywhere in between these regions. I think the limits of human physiology may dictate where the ideal Goldilocks zone is. For example, depressing metabolic rate such that body temperature falls below 28 degrees Celsius may not be possible. Another variable is time. So synthetic torpor could be induced and held for long periods of time or chronically used, as we've seen in so many science fiction movies. Alternatively, it could be used intermittently, perhaps during the biological night, or it could be used in a targeted fashion where crew members are only placed in this metabolic state in response to or in anticipation of some events like a solar particle event or an emergency of some sort. So any of these methods could be relevant to a Mars mission, and theoretically, any combination of time and metabolic rate is possible. Given this, and with respect to the gut microbiota, my guess is that the use of torpor that will have the greatest impact on the microbiota is a deep state of torpor held for a long period of time. And this is because, as has been written about and mentioned earlier, those torpor states may require nourishing the crew member via IV lines or total parenteral nutrition, TPN, which would bypass the GI tract and possibly lead to the microbial changes that we see in our ground squirrels. Now, the thing is, the hibernators have adapted to these conditions, but humans have not. There are differences in the way humans and hibernators respond to a lack of food flowing through the gut, some of which I've highlighted here on this table from a recent review paper. And suffice it to say, these responses may have negative implications on crew member health, so we may want to avoid them. My best guess, for whatever that's worth, is that we should try to maintain the crew members' gut microbiotas for reasons discussed in Trisha's previous workshop on the gut microbiome, so as to optimize crew member health. This would mean ensuring that crew members are nourished through eating and not through TPN. This would also avoid some of the complications that I showed on that table on the previous slide, and avoid the need to, for example, partially rely on microbes to recycle nitrogen during these metabolically depressed states. With respect to synthetic torpor, of course, this would mean holding crew members in this state not chronically, but intermittently, as this would allow them to regularly be in an active metabolic state during which they could eat. But this is, of course, based on our current knowledge, which is limited. The fact is there's an awful lot that we don't know about this. And so I think the upshot is, if and when we start testing whether the numerous theoretical benefits of synthetic torpor to human spaceflight are actually realizable, which I sincerely believe hope they are because they are significant, then we should include investigations into the causes and the consequences of changes to the gut microbiota. Thanks for your attention, and thank you for the invitation to be part of this event.
Thank you, Matt. This was a really, really good presentation, and I think that you were able to connect the dots with our previous microbiome workshop. So that was really good. So I think we have time for one question, and I want to remind everybody that we will reconsider all of the questions that we haven't been able to answer at the end of the workshop. I think you're muted, Gary.
Sorry about that. We'll ask a real quick question first. Do the hibernators maintain urea excretion similar to in hibernating and non-hibernating periods? So is it similar in those two time periods?
In terms of, so they don't urinate, so they're like a closed, they're almost like a closed system, if I understand the question correctly. So urine accumulates in the bladder throughout hibernation, but at a very slow rate.
Okay.
And urea concentrations in the blood plasma are significantly lower during hibernation than during summer active season. That's probably a result of spending so much time at a metabolically depressed state.
One last question here. Do you consider hyperporosity in the gut lumen to be a major source of pathogenesis in human spaceflight?
I guess I would probably have to read more into that to provide a definite answer to that question.
Fair enough.
Sorry to cop out.
No, that's fine. Okay, we'll move on to the next talk then.
Thank you, Matt. Really appreciate it. And we'll go with our next speaker.